Thank you very much for allowing me the opportunity to talk today at this symposium. I'm going to share my screen and talk to you about irisin and osteoclasts and its potential role and its relevance to space flight. So irisin is a mediator of bone resorption. I'm going to first talk about irisin and its relevance for space flight, then talk about some of the genetic models we've made, both global deletion and global overexpression, and then in vitro studies of irisin on osteoclasts with questions for future studies. So why is irisin, which has been considered an anabolic factor for a number of different tissues, relevant for spaceflight? Well, one has to understand the importance of the bone remodeling unit and the relative impact of irisin or any other circulating hormone on bone turnover. And it turns out that the osteocyte is the master level control over bone remodeling. And normally, as you all know, skeletal remodeling is balanced between bone resorption and bone formation. And it's likely that bone resorption begins that process via signals from the osteocyte, possibly through ligand. And that leads to the generation of a enzyme and acid generation to lead to release of growth factors from the skeleton, which then can recruit osteoblasts as well as osteoclasts, generating clastikines that can recruit osteoblasts. They then can act to stimulate osteoclastogenesis or inhibit it, depending on whether these proteins are either RANKL, which is a master regulator of osteoclastogenesis, or OPG, which binds to RANKL and prevents further resorption. So we know spaceflight causes uncoupled bone remodeling, that this process is pathological, that there's an increase in bone resorption and a decrease in bone formation, which leads to rapid rate of bone loss while in space. And this can be measured in a number of different ways. The most sensitive way is measurement of bone resorption with urinary CTX in spaceflight, both in humans and animals. And bone formation can be measured with P1CP or P1NP or osteocalcin, both of which go up, but not to the same extent as bone resorption. And it is important to note that during spaceflight, sclerostin is also increased, and sclerostin is an inhibitor of bone formation. So with that in mind, let's look at irisin and see if we can make any connections between greater rates of bone resorption and irisin itself, or the catabolic effects that occur with spaceflight and the subsequent skeletal changes. So irisin is a myokine. It's produced as a result of cleavage of FMDC5 from skeletal muscle. Irisin circulates in the circulation for a relatively short period of time and increases 1.5 to 2-fold during acute exercise. And its levels are in the range of 3 to 10 nanograms per ml in the circulation. We know that irisin induces thermogenesis, and I'm sure you'll hear more about that in this meeting. And it does so by being cleaved from FMDC5, which is regulated by PGC1 alpha, a major mitochondrial regulator of thermogenesis. And irisin acts to increase mitochondrial oxygen consumption and causes beiging of white adipose tissue. This leads to reduced adiposity and decreased insulin resistance. And this has been conceptually considered one of the future models for understanding how glucose clearance could be mediated by agents that change fat cell behavior. So our lab has been interested in understanding if irisin has a skeletal effect. And the reason we say that is because we have been interested for a long time in understanding the bone metabolism connection. And so targeting a number of different bone cells could impact bone turnover and could play a role in regulating overall metabolism. So the Grano group has been taking the lead on understanding how irisin could regulate bone, and they have a number of different studies that have shown that when irisin is administered intermittently to bone, to mice, it can increase osteoblast differentiation. Its effects on bone mass have been somewhat controversial. So even though trabecular bone is much more sensitive to endocrine hormones because of its fast surface area and high turnover rates, irisin itself does not really impact trabecular bone, but it does appear to have an impact on cortical bone. And this might lead one to believe that irisin secreted from muscle, which attaches to bone, would have a direct paracrine effect on the skeleton. And so their studies from the Grano group have shown that that this, in fact, can be the case. They also administered irisin to unloaded animals and found that they could prevent some of the bone loss that occurs with skeletal unloading by the use of intermittent doses of irisin subcutaneously. This is the data on loading and unloading in the mouse experiments published by Grano, suggesting that there is an impact of unloading on resorption when treated with irisin in the red bar on the left, and on the right bar, you can see that unloading increases bone resorption, although it's not detected in their studies by increasing rankle in the marrow. But you can also see that resorption is further increased by treatment with irisin. So we were fascinated by both the intermittent nature of irisin administration, which might have some potential benefits if it's anabolic to the skeleton for future spaceflight, and the fact that there was an increase in bone resorption, which could lead to a triggering of increased bone remodeling and ultimately maybe a positive net effect on bone. So I should remind you that other factors are involved with skeletal unloading, and that includes sclerostin and DKK1. And this increase in sclerostin is fairly uniform across unloading experiments, both in mice and humans. So we decided to take a genetic approach instead of using recombinant irisin, which has its own issues related to how it's derived. And we decided to look at both the transgenic and conditional and global deletion of FMDC5, the parent peptide. And we were surprised. We used a targeted approach using a muscle-specific FMDC5 transgenic made by Eric Olson at the University of Texas Southwestern. And what we found was that despite our preconceived hypothesis that an irisin would be anabolic to bone, we found a rather dramatic lessening of trabecular bone in both the femur and in the vertebrae, as shown here. In addition, when we looked at the data, particularly in younger animals, there was a marked reduction in bone volume fraction and a reduction in trabecular thickness, number, and spacing. Moreover, bone formation rates were low and mineral apposition rates were low. And the eroded surface in the number of osteoclasts per bone perimeter was increased, although it did not reach statistical significance. So what did this tell us? It suggested to us that contrary to our a priori hypothesis, irisin actually was driving enhanced bone turnover and possibly a net loss of bone. Thanks to Roland Barone at the Harvard School of Dental Medicine, we did backscatter electron microscopy to try to understand whether part of that is driven by increased osteocytic induction with sclerostin and osteocytic osteolysis. And here you can see in the transgenic mouse at 8 weeks, much more so than at 13 months, there is this marked increase in the number of lacunae and the number of empty lacunae, so that lacunar density is increased, suggesting that in fact osteocytes are driving some of the changes that we noticed in bone. When we looked at biochemical markers of bone turnover, bone formation was reduced at 4 months, everything neutralized by 13 months of age, but you can see that both P1NP and osteocalcin measures of bone formation were reduced to a degree similar to what we saw by histomorphometry. And that may be related to sclerostin, as you see in the upper right hand corner, which went up about 50%. And this is very consistent with what we see with unloading, this increase in sclerostin. Unfortunately, the FNDC5 conditional transgenic, the targeted transgenic, actually is a little leaky. And when we looked at bone marrow FNDC5, we found that even though it's a muscle-specific promoter, that there were significant amounts of FNDC5 in the marrow. So we can say that the transgenic has a defect in bone turnover, probably accelerated bone turnover, although we can't say that it's absolutely specific to irisin from muscle alone. About the same time that we were studying those mice, Dr. Kim noted that when we treated osteocyte cell lines, MLO-Y4, or osteocytes isolated in vivo from intact animals, that irisin caused a dose-dependent increase in sclerostin, consistent with what we saw, that is the decreased bone formation and the increase in bone resorption, which is an indirect effect of sclerostin mediating rankle production. The other provocative thing about the FNDC5 transgenic was the number of osteoclasts that were isolated in vitro from primary animals. And here you can see that in 3 different sets of experiments, there were many more osteoclasts that were bigger in the transgenic than in the wild type mouse. So muscle-specific overexpression of FNDC5 in vivo, decreased bone formation, increased osteoclast number, leaky promoter, giant osteoclasts. There were more of them. So we then turn to deleting FNDC5 and asking the question, is this protective or is it deleterious for the skeleton? And here you can see in our FNDC5 knockout mice that there's a marked increase in trabecular bone volume fraction and an increase in trabecular bone density and connectivity density, as well as a reduction in SMI, all consistent with a greater increase in bone mass in these animals, primarily in females, not so much in males. When we did ovariectomy in the older mice, what we found was that there was this decrease in bone mass that occurred in the wild type mice in blue, but in the orange, these mice were protected from ovariectomy, which is quite unusual, but consistent with the possibility that these mice, in the absence of irisin, actually had less bone resorption, and which is the major feature of ovariectomy-induced bone loss. And in fact, that is the case when we look histomorphometrically. The osteoclast surface and number of osteoclasts, which are markedly increased in the wild type, did not change in the FNDC5 null mice. In addition, there were less osteocytic lacunae, and even with ovariectomy, there appeared to be less. So we then asked the question, what is the mechanism of this difference in bone turnover, where you get the absence of irisin associated with protection versus the overexpression, which causes bone loss? And we focused on the osteoclast based on our data about CTX and sclerostin. And as I remind you, Dr. Kim had already identified that sclerostin is a downstream target of irisin. In fact, when we cultured primary bone marrow cells from black 6 female mice, we were able to show that with the addition of irisin at 2, 5, and 10 nanograms per ml, which is near or on the physiologic range, there was a marked increase in osteoclast number per well, and a marked increase in alpha V beta 5 expression, which we suspected was the major receptor for irisin. And this was repeated several times. We also treated these cells with anti-IG integrin alpha V beta 5 antibodies, and we're able to show that once those cells were treated with antibodies, the increase in osteoclastogenesis with irisin 10 nanograms per ml was blunted. So we then did a dose response curve in the upper right-hand corner, a time response curve, sorry, in which irisin was incubated at 4 hours, 24 hours, and 7 days in an attempt to try to determine whether this is intermittent irisin release during exercise or more chronic sustained release of irisin. And what we found was the highest resorption really occurred with the longest time of exposure, 7 days. What was interesting, though, was that the number of osteoclast nuclei per cell was increased markedly during differentiation in those cells treated with irisin versus control. So there was an overrepresentation of cells that had nuclei of 25 or more compared to controls. And you can see in the bottom left the difference in the nuclei and the size of the osteoclasts. So there was increased size, and the number of nuclei were greater, suggesting that irisin could modulate fusion of osteoclasts. But we were still concerned that we didn't know whether that represented a change in bone resorption or whether this was true and unrelated to the higher rates of bone resorption we saw in the overexpressing model. So we did some in vitro resorption assays using both dentin and commercial assays, the osteo assay, and you can see in the bottom left that irisin increased the number of those white spots that were consistent with bone resorption occurring osteoclasts on the surface of the artificial bone caused resorption. We also were concerned that this was artificial bone surface, so we cultured osteoclasts and treated with irisin in those surfaces in which there was dentin that we received from Dr. Barone's lab. So native bone, you see a very similar increase in bone resorption. Finally, we did a Corning resorption assay and a Lanza resorption assay just to prove definitively that the resorption was related to irisin treatment. And here you can see that with the Corning assay, there's this increase, and this is more pronounced at 7 days and driven by increased osteoclast number. So the next thing we did is an unbiased approach to ask the question, so what happens to the signaling mechanisms in osteoclasts in response to irisin? And here you can see a volcano plot and the RNA-seq evaluation with hierarchical clustering. And you can see the controls are down here, the irisin is up here by PCA, consistent with the differential effect. And here you can see how in vitro RT-qPCR of those differences were confirmed, including genes related to fusion as well as differentiation. And it's of note that the rank, which is the rankle receptor, is highly expressed on osteoclasts and is increased by irisin. This would be consistent with an additional role for irisin in supporting bone resorption and osteoclast differentiation, in addition to its effects on from irisin, but increasing rank expression, which allows the rankle in the conditioned media to stimulate further osteoclast-mediated bone resorption. And as you can see, the resorption markers are markedly increased. and several wints are induced by irisin, and we suspect these are clasticons that can drive osteoblast-mediated differentiation, which osteoblast differentiation and bone formation, which would be consistent with some of the work from Grano et al. And here's a list of some of the RNA-seq data, downregulated and upregulated genes. There were 26 genes that were downregulated and 28 that were upregulated. Most of them were osteoclast differentiation or fusion genes that were noted. So what are the potential targets for irisin and osteoclasts? Well, we think that it does upregulate rank, which makes the osteoclast more sensitive to endogenous rankle. But we also think that the VATPases may be increased by rankle, by irisin, as noted in our our RNA-seq experiments. And there may be some changes in the lysosome that we're still looking at in terms of release of degradative enzymes. So let me summarize my talk by gain and loss of function studies with FNDC5. Forced expression of FNDC5 causes bone loss, reduced bone formation, increased sclerostin, and greater resorption due to increased osteoclast number. In vitro studies of the transgenic showed larger and greater number of osteoclasts. Deletion of FNDC5 is the opposite. It results in protection against OVX-induced bone loss. And irisin upregulates sclerostin and rankle in osteocytes and drives up osteoclastogenesis. And I should say also rank. And irisin activates the alpha V beta 3 integrins on osteocytes and osteoclasts. So what are our challenges moving forward? Well, first we have to understand what is the enzyme responsible for the cleavage of FNDC5 to produce irisin. And we need to know where circulating irisin is coming from. It's clearly coming from muscle, but there are other possible components, including heart and other tissues, including adipose tissue. So those are going to be addressed by conditionally deleting FNDC5 in a tissue-specific manner, and those mice have recently been made in the Rosen lab. We also want to know if CD81 is a co-receptor for integrin receptors, or does it work separately but integrates signaling with the osteoclast? And what are the clasticines that are induced by irisin, and does that impact bone formation? And we're still at the early stages of understanding in co-culture how osteoclasts can drive osteoblast differentiation. So with that, this is the Spiegelman lab, this is the Rosen lab, and this is the support from NIH. Thank you very much.